Okay, thank you all for coming. Today I'm going to tell you about beyond vision, how light affects our behavior. And I know Manuel had an issue and may not be here, but I would like to thank him for the invitation. And I'm really excited. I see a lot of people I know online, so I'm really excited. Hopefully, I'll see you guys in SRBR soon. Before I start my talk, I would like to acknowledge the people who helped me do the work. We were Mouse Trilab, where myself, I can't find myself here. Oh, here, I was staying here. Myself, Hai Chinzao and Reggie Provilla, three mouse neuroscientists together, we were sharing resources, reagents, and ideas, which allowed us to go beyond our little area of expertise. And then, as mentioned by the introduction, in 2017, I moved to the National Institute of Mental Health. And these are the current members of my lab. And I will be acknowledging people who did the work throughout my talk. So because of the rotation of the Earth around its axis, something really exciting happens. So if you look at 1 centimeter square of your skin, in a sunny day without any clouds, you can get 10 to the 6 photons per centimeter square. And this is not a typo. Actually, this is 10 to the 16 photon per centimeter square per second. But the same 1 centimeter square at night can give you only a few hundred photons, especially if you don't have moons and you know it's cloudy, for example. So it's really interesting that this allowed animals to know how to differentiate between day and night, but it produced a major problem for vision. Vision now has to be able to be functional across all this different spectrum of these light intensities. So interesting, in mammals, the retina, just like other vertebrates, have been the area where rods and cones and retinal circuits have been important for visual functions. But interestingly, in vertebrates, there was other non-ocular photoreceptors. So this is their eye, but there were other non-ocular photoreceptors, mostly in the brain. So we know that the pineal, for example, is photosensitive, but there's also deep brain photoreceptors. And I know most of you here, I've seen names that they kind of appreciate the importance of light and circadian rhythm. But I love this work from Yoshimura's lab in Japan, where you're only changing the amount of white light that you're giving to the birds. So all you're doing here is, if you're presenting a short day versus long day, what you notice is that the gonad size of the birds changed tremendously. And this change in gonad size is really interesting because it allows the animals to actually mate across the season. So light is not only important for our circadian rhythms, but it's also important for reproductive function across the life history of the animal. However, in mammals, it seems like all light detection have been centralized to the eyes. And at least the pineal, for example, and other deep brain photoreceptors have not been shown to be important for major physiological functions that I'm going to be talking about today. So how do we detect light? It's actually quite simple. In mammals, light has to cross the pupil, and it has to hit the back of the eye where the retina, the light-sensitive organ, is. The retina forms the optic nerve via the axons of retinal ganglion cells and project to many brain regions. For you to appreciate this beautiful image here, light has to hit the lateral geniculate nucleus of the thalamus, and for movement, it has to go to the superior colliculus as well as other accessory optic area for you to view this image and track the object. So you're consciously aware of this image when you look at it here. But light also detects, and as I told you, I'm sorry, I jumped ahead of myself. And that happens because, as I told you before, the retina has these beautiful photoreceptors, rods and cones, that allow you to take the photon energy and transfer it to an electrical signals through phototransduction pathway. So let me give you a little primer about the retina. So as I told you, light has to cross the pupil. And light actually comes from this direction. It has to cross the ganglion cells, of which, as I told you, the axons make the optic nerve. And then it has to cross many neurons in here, even the initial segments of rods and cones, before it hits the photosensitive parts of rods and cones that contain rhodopsin and conopsins. And what is really remarkable is, for example, rods are so well evolutionarily designed that they can detect a single photon. And just to remind you that in the day, you could have 10 to the 16 photons per centimeter square per second on your skin. So the big question is, the retina is known to also affect other behaviors, such as circadian photoentrainment through mostly the suprachiasmatic nucleus. And if you ask me questions, I'll tell you a little bit about both the intergeniculate leaflet and now we think the ventral part of the lateral geniculate nucleus. And again, for this audience, circadian rhythms are really important because they allow you to change across the day-night cycle. So for example, at 6 a.m. in the morning, you have the sharpest rise of blood pressure, and this is where strokes and heart attacks happens mostly. But I want to spend a little bit of time and tell you about a very simple reflexive behavior known as the pupillary light reflex. So this behavior is very simple, really. It's literally the pupil opens in darkness and closes in light. And honestly, this behavior could have easily been driven by having a photopigment expressed in the iris muscle itself. So you don't even need the retina. You could just express a photopigment in the iris muscle that can locally drive the pupil constriction. In fact, some vertebrate animals have that. They have actually a pigment in the iris muscle that drives constriction. However, in mammals, this behavior is very complex. It actually has to grow, the light has to go first through the pupil itself, hits the retina, and then goes to an area in the brain called a liver pretectal nucleus, and then through multiple synapses come back to the pupillary muscles and drive pupil constriction. So why do you have such a complex circuit for supposedly a very simple behavior? So what I want you to know by the end of my talk, hopefully today, is that we know that light detection for image functions is complicated. But what I would try to convince you by the end of my talk today is that even light detection for non-image forming functions is very complicated and varies across behaviors. And I will show you in details how we discovered this. So how do we model circadian rhythm in the lab? It's quite easy. Circadian field has been amazing at this. These behaviors are very, very quantitative. So we simply put a single mouse in a cage and we measure their activity across multiple days. And we double plot the data and we change the light environment. So the first 10 days, you have 12 hours of light, 12 hours of dark. And mice are nocturnal animals, so most of their activity are confined to the dark portion. And if they photo-entrain to the light-dark cycle, they have an exact 24-hour period. However, when you put them in constant conditions, what you could see is that now their period is not 24 hours. In this case, it's 23.8, and it runs only by the endogenous circadian pacemaker located in the SCM. So this is an endogenously driven circadian rhythm. So what light does, it makes this inaccurate circadian clock very physiologically relevant so the animal can survive in the environment. So as I told you, the retina has these beautiful photoreceptors, rods and cones. So for many years, there was a lot of fighting in the non-image fields of whether rods or cones contribute to circadian photoentrainment. But really, these two beautiful papers from Russell Foster's lab were published in Science back to back, even though they didn't discover the photoreceptors. So that's how surprising these results were. So in these studies, the authors produced an animal that, oh, by the way, when I put the references on the left, it means somebody else did the work. When I put them on the right, it means my lab did the work. So they decided to create animals that completely lack rods and cones, hence the name rodless coneless. So they destroyed all the rods and cones genetically. And what they found remarkably is that wild-type animals and rodless coneless animals photo-entrain not only at high light intensities, but also at low light intensities. So this told them that there has to be some photoreceptors that are found somewhere else apart from the photoreceptor layer that allows that retina to compensate for the absence of these photoreceptors. Because if they remove the eyes from these animals, then these animals will free run. They will not photo-entrain to the light dark side. So to make a long story short, that was 1999 was my last year of my PhD. And I was really lucky to follow the pioneering work of Ignacio Provencio and David Burson, and then discover with them that there is a subset of ganglion cells, the cells that actually project directly to the brain, that are intrinsically photosensitive. That's a fancy way of saying that they are photoreceptors, because they express a photopigment similar to the rhodopsin and conopsin, which Ignacio Provencio has called melanopsin. So I'm going to refer to these cells throughout my talk as intrinsically photosensitive retinal ganglion cells, or IPRGCs. Okay, so I know I talked a lot, so I just want to quickly summarize that we now know that the mammalian retina has two types of photoreceptors. We have the classical photoreceptors that are beautifully evolutionary evolved to detect even single photon in the case of rods. And we have these nondescript photoreceptors that are neuronal in structure because they are ganglion cells similar to the retinal ganglion cells that could detect light intrinsically through the melanopsin system, but they also incorporate rod and cone light input similar to the other conventional ganglion cells. So they have a dual personality. They have an intrinsic response driven by the phototransduction of melanopsin, and they have an extrinsic response driven by the retinal circuit through rod and cone. So the question is, what happens if we knock out melanopsin? So we knocked out the melanopsin protein and animals photoentrained. So we wondered if there's still more surprises in the retina. However, there wasn't any, because if you knock out the phototransduction pathway of rods, cones, and IPRGCs, and you produce in triple knockout animals, now you could see that these triple knockout animals are unable to photoentrain to the light outside. So this is quite remarkable because this is consistent with what we see in a human from the beautiful work of Chuck Seisler, that shows that if there are two types of humans, there are blind humans that can photoentrain because they probably have the IPRGCs functional. And then you have blind humans that have cyclical sleep problems because they free run throughout the light-dark cycle. Okay, so as I told you, rods and cones can signal through the melanopsin pathway. And rods and cones are beautifully evolved to detect, even in the case of rods, I repeated the third time, single photon responses. So why would you need melanopsin to be preserved evolutionarily? In fact, if we look at all vertebrates that express melanopsin, not only melanopsin is preserved in these vertebrates, but it's also expressed in what seems to be a subset of retinal ganglion cells. So there must be something evolutionarily important for melanopsin that the rods and cones cannot compensate for. And this idea actually kind of really worried me a lot, because this work in 2007 by Kun Wan, when he was in David Berson's lab, showed not only that rods and cones can signal through the IPRGCs, but in fact, they make them better. So if you look at the control, these are IPRGCs that have rod and cone input, and these are without. What you can see directly is that IPRGC with rod and cone input are much more sensitive to light than the ones without. And second, they are more better responders. They respond on the onset and offset of light, while these cells take a long time to respond. So this worried me, is like, is melanopsin then there for, and as I told you before, melanopsin knockout animals photoentrain just as nicely as wild type animals under 12 hours of light, 12 hours of dark. So we got a hint that melanopsin is actually more important than that in a sleep paper that we published in 2008. And now what I'm showing you, instead of actograms, I'm showing you somnograms, where the flat lines are non-REM sleep and the spikes are REM sleep. And as I told you, mice are nocturnal animals, so they sleep mostly in the day, but as you can see very nicely, they also sleep at night. Actually, mice sleep in a very interrupted fashion, and they sleep approximately 50%, including the day-night cycle. So what we decided to do, we wanted to ask, it's nice to study photoentrainment to 12-12 light-dark cycle, but what if we come 2 hours after darkness and give the animal a bright light pulse? And what we notice in the case of wild-type animals, that just you come in the middle of the night and you shine light and you directly induce non-REM and REM sleep. There is a clear effect on non-REM and REM sleep. So what happens in melanopsin knockout animals? So we know melanopsin knockout animals photoentrain to this cycle, so their sleep photoentrains. Now what happens if, and we think this is because, as I told you, rods and cones can signal through them. Now what happens, although they can photoentrain, they cannot actually respond to this direct light effect on sleep. So there seemed to be a very important function for melanopsin, not for circadian photoentrainment per se, but for the direct effect of light on behavior. And you could see this much nicer if you look at body temperature. So this is work by Alan Roop, who was in the lab at the time. And what you could see is that this is the body temperature of the mouse, and the body temperature is high when the mouse is active, and it goes down with sleep. And again, Alan did what Kara did, and he came 2 hours after the dark onset and gave 3 hours of light pulse. And you see that light can directly and continuously lower body temperature in the mice. And you could see the expansion here of that effect on body temperature. Now, what happens in melanopsin knockout animals, as you would expect? These animals beautifully photoentrain to the light-dark cycle, so their body temperature have a beautiful daily rhythm. But now when you come in the middle of the night and you shine light, now light is incapable of driving changes in body temperature. So that was really interesting, but how many people go in the wild and 2 hours after darkness, they get a very bright light pulse? So luckily for me, Melissa Semen saw this data. By the way, Melissa has her own lab now at Oakwood College, saw this data, and she decided to ask question, maybe this light pulse indicates some form of seasonality. So the only thing she did is she put melanopsin knockout animals under different light-dark conditions, and this is unpublished yet. So she put wild-type animals and melanopsin knockout animals in 12-12 light-dark cycle, and as many labs that we sent the mouse to showed, these animals beautifully photoentrain under 12-12, similar to the wild-type animals. But here's the shocking result. Now, if you just change the amount of light in the day by 4 hours, so instead of 12-12, you give 16-8, wild-type animals still do it beautifully, but now melanopsin knockout animals start having serious problems in photoentrainment. So this told us that although rods and cones can compensate for the absence of the melanopsin protein under 12-12 light-dark conditions, under different seasonal light treatments, it seems that rods and cones failed completely, not completely because there's still some entrainment, but failed miserably at photoentraining the animal in the absence of the melanopsin protein. And this was very satisfying because it showed a really important physiological function for melanopsin for measuring seasonal effect of light. So let's go back to the cells. So in 2002, as I told you, I was involved in labeling these cells and tracing them to the brain. So here we labeled IPRGC, and now we know we're only labeling one subpopulation of IPRGCs, which is the M1. So this is only the M1 population, and you can see very beautifully it's projecting to the SCN, as you could see here very nicely. The SCN is, actually, you don't see it. Let me just 1 second let you see that. As you can see here very nicely, this beautiful innervation of the SCN from this region. Okay. So when we first discovered them, we thought that these are circadian photoreceptors. To quickly in my, okay, now I have to go back to the word. This is to quickly discover that they are not only circadian, but they have much more general effect. They project to sleep and alerting center. Not surprisingly, they project to the pupil centers, and they also project to mood regulating centers in a region that we recently discovered we call the perihabeneal nucleus, which I'm going to tell you about later. And because of these 3 amazing women in the lab, we now know that IPRGCs are at least 5 or even 6 different subtypes. Tara Legates and Tiffany Schmitz have their own labs right now. And Tiffany in her own lab showed that these M4 populations are no other than the classical ganglion cells that detect contrast, and they are the unsustained alpha ganglion cells, which are also intrinsically photosensitive. So if you have any questions, I'll be happy to answer more questions about the different subtypes. But even before we knew about the subtypes, Ali Gulair, who also has his own lab now at UVA, decided to ask a very simple question. We know if we knock out melanopsin that at 12-12, the animals can photoentrain very nicely, but what happens if you knock out the cells? So this is actually, we've done these experiments in 2008, before even the discovery of the multiple subtype of IPRGCs. And talking to Jeremy Nathans, he suggested that we should use the attenuated diphtheria toxin because the diphtheria toxin may be too strong and doesn't allow us to get germline transmission. And that was very lucky for us because when we used the attenuated diphtheria toxin, we were only able to kill the M1 IPRGCs. Clearly, you're not going to kill the conventional ganglion cells because they don't express melanopsin. But we also didn't kill the non-M1 IPRGC we knew later after the publication. Importantly, we actually have supplementary data that showing that even M1 IPRGCs died only 2 to 4 months after birth. So because they only died in adult stages, vision, image forming function, development, and functionality were not affected. So we were very lucky in a way that we produced a conditional knockout animals without our knowledge. And that animal is the opposite of the rodless coneless animals that Russell Foster group has produced, where it seems to have normal image forming functions, but now we could ask the question, what happens to non-image forming visual functions? And honestly, this has been 16 years in the making, and every time I see these actograms, I'm like in shock of how clean these animals are, because they exactly look like the triple knockout animals. In fact, again, the wild type controls beautifully entrained, while these ADT animals completely free run throughout the light dark cycle. So this told us very nicely that two things actually. One, that there is a evolutionary diversion between image and non-image forming visual functions at the level of the retina and specifically at the level of the M1 IPRGCs. And second, that because of these animals, we now can study circadian disruptions in the context of a normal image forming functions. So we could actually separate the effects on circadian functions versus image forming functions. Okay, even more lucky for us in a way, Alan Chen, who has his own lab now at National Taiwan University, I'll give you the punchline and then show you the data, did intersectional genetics where he mated the melanopsin Cree line with brain3B log speed stop DTA. So to give you the punchline, only cells that have these two promoters active at some point of their lifetime will remove this stop area and express DTA in IPRGCs. And what he found, and I'll show you the data, he found that only SCN projecting M1 cells are brain3B negative, while the majority of M1 cells that project to non-SCN regions and all non-M1 IPRGCs are brain3B positive. And so what this allowed us to do, allowed us to ablate all the non-SCN projecting IPRGCs without affecting the SCN projection. And to show you this in a very nice way, we labeled all ganglion cells now conventional and IPRGCs with chlorotoxin. And what you could see here is that the image forming centers are still labeled in both lines, although there is a reduction in the M4 because the M4 are dead in these animals. The SCN is beautifully innervated. There is a little bit of reduction as well, but not much. But if you look at non-SCN regions, they are completely denervated. And consistent with the fact that the SCN is still innervated, these animals were able to photoentrain similar to the wild type animals. That despite the fact that they have between 200 to 400 IPRGCs per retina. So this again told us not only M1 cells are necessary, but they are also sufficient for circadian photoentrainment in the presence of melanomas. So now we think that this SCN oscillator that is so important for regulating the circadian timing system in humans really receive input from very few ganglion cells in the retina, the M1 IPRGCs. So such an important function receive all its light information from the environment through a very small subpopulation of ganglion cells. And this is quite remarkable to me because we know how important this system is for the survival of the animals. Yet few cells are capable to allow the animals to photoentrain to the light dark environment. But this also gave us something really interesting. We wanted to ask, remember how I showed you that melanopsin knockout animals do not respond to the direct light effect on behavior? So we wondered if this body temperature effect of light also depended on the SCN or not. So now we use the melanopsin Cree line as control because we have to use Cree to eliminate the brain3B positive IPRGCs. And just like the wild type animals, they have a beautiful rhythm and they respond very nicely to a 3-hour light pulse in the night. But now, surprisingly, look what happened at the animals that have a beautiful SCN connectivity. They photoentrain, as I told you, they are sufficient for photoentrainment. But again, now similar to melanopsin knockout animals, they have no response to this direct light effect at body temperature. So that told us that something different than these SCN projecting neurons somewhere in the non-M1 SCN projecting IPRGCs that drive this response. This also allowed us to come up with a, honestly, at the time, a courageous idea that we think there are two different pathways. And by the way, this direct pathway has been called masking in the past, which I think was a very unfortunate name, although it was important for the times. So we think that in addition to this circadian photoentrainment pathway, there is this direct pathway where light can affect behavior independent of circadian photoentrainment. By the way, my lab is still very interested in this and understanding this pathway, and half of the lab is still working on this, and I'll be happy to discuss this with you if you're interested. But our goal for this part was to reveal the circuits and mechanism by which light directly affect behaviors. And the reason I'm putting these behaviors here because we've already shown some evidence that light can directly affect these behaviors. So I'm going to show you the punchline and then take you through multiple behaviors that we showed where light can directly affect them. So first of all, we found that light can directly affect cognitive functions through the SCN, actually surprising, but in a clock-independent fashion, through yet another not known brain regions to affect the hippocampus in learning and memory. We found in 2020, Diego Fernandez, who also has his own lab right now, that developmentally, the IGL is required for setting up a time feeding mechanism to allow time-restricted feeding through the SCN. I'll show you some of that data soon. And in 2022, I think, I can't see the date. Let me just make sure I can see the date. Yeah, 21, sorry. In 2021, we showed that a subset of IPRGCs project to the pre-optic area, which then project to wake alerting centers in the brain to affect specifically non-REM sleep. Actually, it has no effect on REM sleep or body temperature. Okay, now I have to go back. And I'm going to tell you a lot about the PHB, the perihabinular region, that project to these regions to affect mood. This is going to be a big part of the remaining part of my talk. And recently, we just published a paper just recently showing that this PHB is also important for the medial prefrontal cortex function and for social behavior. So now we know that the circadian photoentrainment pathway is very important, but as I told you at the beginning of my talk, light reception for non-image forming functions is quite complex and involve both the circadian photoentrainment pathway and this direct pathway that doesn't require the involvement of the SCN pacemaker function. So because of that, we decided to come up with a new hypothesis in the lab, which we call the tripartite model, where as a circadian biology trained person, we always thought that light-mediated behavior are driven by homeostatic drive, but by the circadian drive, and all light environmental effect has to go through the circadian pacemaker to affect behavior. But now we think there is also a completely separate arm of this pathway, where light can directly affect behaviors, which is the majority of our work, but it also could affect the homeostatic drive itself, and we haven't found clear evidence yet for that in our experiments. And really, the importance of light-mediated effect on behavior will only be produced through both the circadian and the homeostatic, as well as the direct effect on behavior. Hence the name tripartite model. So let me show you, for example, how we think of this for the case of feeding and sleep. So let's start with feeding. There's no doubt that the arcuate nucleus is so important for senses, both hunger and energy level. That's the region for feeding. But that the SCN is also important for when do you eat, the daily regulation of food intake. So honestly, we thought one of these two regions would be important when we look at time-restricted feeding. And again, this is the work of Diego Fernandez, who has his own lab right now, and Ruchi Kumar, who's a wonderful postdoc still in the lab. And so what they did, they gave animals constant darkness, and they gave them food ad libitum here, and then they restricted the food to 7-hour levels, and they measured the food anticipation before the presence of food. And what you could see here, if you look at the feeding rhythm, you could see that animals still have a nice clock in constant conditions. They feed with a close to, but not exactly 24-hour period. But now, when they anticipate, when you give them food, you just eat it when you give them the food as you would expect it. But what you notice here is their activity rhythm is different. So here's the rhythm in DD. And now what you see this beautiful anticipatory response, which in correlation with food, you could see that they are anticipating the presence of food.
Mm.
And so again, we thought this is gonna require areas that have been shown to be important before for this function, but we were actually quite shocked when we used animals that lack IPRGCs during development. So now this is an melanopsin-driven animals that drive full-strength DTA, and the full-strength DTA kill IPRGCs early in development. And what we noticed is that this animal is, as you know, completely incapable of photo-entrainment to the light-dark cycle, but remarkably, it was also unable to entrain to the time-restricted feeding. Even when its rhythm would fall at the same phase as the anticipation, this animal would not have the same anticipation as the wild-type animal shown here. And here's the quantification of the data. So this paper is published, so I'm going to give you the punchline, and I'll be happy to discuss it later. So what we think is going on is that there is a food entrainable oscillator, which we still don't know where it is, that senses the food scarcity, and then through input to part of the LGN, part of the visual thalamus, the intergeniculate leaflet, and through MPY cells in the IGL, inhibits the SCN. And this allows the animal to actually start being active at times when they are not supposed to be active. What is really interesting is this whole system here requires the IPRGCs to be set up. So it seems that in the case of non-image forming visual functions, the absence of photic entrainment doesn't enhance responses to non-photic stimuli. It actually decreases it, which is quite different from the sensory system, where blind humans usually have better touch and better hearing. The other process that I would like to express the tripartite for you is the sleep. So we know for many years, sleep researchers know this much more than me, that the sleep is regulated by two components, circadian and homeostatic. The circadian tells you when to sleep and is regulated by the light-dark cycle through the SCN. And the homeostatic drives simply how much you're awake you need to sleep to get rid of that homeostatic drive. And again, work from Harris Zhang and Corinne Bayer. Harris still in the lab, and Corinne now has her own job in DARPA, have shown that a very small subset of ganglion cells, IPRGCs that are brain 3B positive, project to a very specific population in the preoptic area, the corticosteroid releasing hormone neurons, which inhibit wake stimulating centers of the brain. So this was really a very smart experiment that they have done. So what they did, they injected a retrograde virus in the preoptic area. And this virus will only express the activating dread in the presence of both CRE and FLIP. So the dread will only be expressed if the cell expresses CRE and FLIP. So they used the melanopsin CRE line, and then they injected in the retina a flipase that will infect all ganglion cells. So only melanopsin cells will express both CRE and FLIP if they are labeled from the preoptic area. And what they found is they labeled between 25 to 40 cells in the retina, and they all correspond to the M1 population. But here's the wonderful experiment. Now, if you put animals under light-dark conditions and you come and activate it with CNO for animals that labeled IPRGCs retrogradely from the preoptic area, what you notice, there is a very beautiful induction of non-REM sleep, but not REM sleep. So this effect is very specific on non-REM sleep. And even though CNO stays longer in the blood, you could see that the effects are transient. And we hypothesize that this is partly because the retina only projects to one population in the preoptic area that controls sleep. So then sleep, again, takes over and restores the amount of sleep to that. So that was really all interesting that there is this direct pathway. So we also wanted to ask, what about learning and memory and mood? And one of the problems of learning and memory and mood is they are usually disrupted by irregular light conditions. So how do you then separate the effect on the circadian clock from the direct light effect on behavior? So again, these are Kara and Tara who did this work. And when they were in the lab, I drove them nuts because I called Kara Tara and Tara Kara, which is pretty annoying. And what they did, they used this cycle that is very well known in the circadian field, but they did something smart in that they removed the wheel. So all the data I showed you before, it's 12 hours of light, 12 hours of dark, or T24. This is a T7 cycle, or 3.5 hours of dark, 3.5 hours of light, repeated for 2 weeks. And again, I'll give you the punchline and then show you the data. When you put the animals in this cycle without a wheel, you don't get sleep deprivation and you don't get circadian arrhythmicity. For example, here you could see that the circadian clock is still running, albeit with a longer period, but it's not arrhythmic. And in the 2008 paper I showed you before, we measured sleep, and the architecture and the amount of sleep are not affected in the T7 cycle. So this will allow us to finally ask a very simple question of whether light can directly affect mood and learning independent of circadian and sleep problems. So what Tara and Kara did, they purchased animals from Jackson's and they put them under either T24 and T7, and they measured the behavior that mimics depression in a human, which is the lack of pleasure-seeking behavior. So what you do, you give mice 2 bottles of water, one spiked with sucrose and one is only water. And mice, if they are like us, they like sugary stuff, they'll drink nearly 70 to 80% of the time from the sucrose spiked water. While the T7 animals indicative of depression-like behavior, you could see their hedonic responses are reduced and they produce what is known as anhedonia. We also did many hippocampal dependent learning and memory tests, including Morris water maze and long-term potentiation measurement in the hippocampal slices. But I'm just going to show you one behavior here, which is short-term novel object recognition test, where you put a mouse in a cage with 2 familiar objects, and then you change one of the object after 1 hour delay, and you give them a novel object. And mice are curious just like us. So if they remember seeing this object, they spend 7 of the 10 minutes investigating this compared to this. And the T7 mice indicative of having learning and memory problem, hippocampal dependent learning and memory problem, they spend the same amount of time with the 2 objects. So this allowed us to come up with a new hypothesis. We clearly believe, because we're circadian and sleep researchers, that sleep and circadian problems can induce mood and learning and memory problems. But we think there is also this amazing direct pathway by which light through IPRGC affect mood and learning and memory. And I'm saying through IPRGCs because we show if you use the ADTA animals that lack M1 in the adult situation, then the T7 cycle did not cause any learning and mood problems. But what's remarkable then, it tells us that image-forming pathways, at least in rodents, do not play a role in driving the light effect on mood and learning and memory. And that is quite interesting, because people, before the beautiful work of Anna Worth Justice and other on the seasonal affective disorder, have said that this usually more subconscious in nature. However, we have to be careful that this is in rodents, and maybe in humans, image-forming pathway might still have bigger function in mood and learning and memory. So then we wanted to ask the question, as I told you, whether the SCN is involved. And again, this is the work of Diego Fernandez here. And who use the SCN, the animals that have only SCN projection. And to our surprise, and believe me, these are true number of animals because we wanted to be 100% sure, the SCN projecting animals, SCN only projecting animals did not show any depressive-like behavior under the T7 cycle compared to the control. That is despite the fact that they still showed some learning and memory deficit compared to the controls. So we actually modified our behavior, our model, and we now think that actually for learning and memory, you require the brain to be negative IPRGC through an SCN independent clock to affect learning and memory. While for mood, you require this region, the perihabenular nucleus, and recently we published in a clock-dependent manner to affect mood. And this is quite remarkable because this region is finally the region, at least in rodents, we haven't yet found it in primates or humans, that now there is a brain region that is a detector of the ambient light in the environment to affect mood. And what's really interesting is that this region, this perihabenular region, which we now know is part of the dorsal thalamus, project to the nucleus incumbens, the dorsal medial striatum, areas known for addiction and reward, but as well as the infralimbic and prelimbic cortices, part of the ventral medial prefrontal cortex, which are important for social behaviors and personality. So when we saw this, we got very excited because we wondered whether the IPRGCs can also play a role in social behavior. And the reason we got excited about that, because of this story of Phineas Gage, who was a railroad worker in the US, and this railroad rod went through his eye orbit and destroyed most of his prefrontal cortex. And as you would imagine, his sleep, his wake, his feeding were not affected because they are not cortical dependent. But what people noticed is that this person was very nice before the accident and was able to keep jobs and no problem keeping his job for a long period of time. But after the accident, he became more socially awkward and more abrupt, and he couldn't keep his jobs anymore. So Lorenzo Esprit at the time in the lab decided to ask a question: does the medial prefrontal cortex is affected by the absence of innervation of IPRGCs to the prehabenular region? And the reason he thought about that, because these neurons, the pyramidal neurons of layer 3 and 4 in the ventral medial prefrontal cortex, or what I'm going to call VMPFC, they are very sensitive to circadian disalignment and stress. Their apical dendrite really changes complexity when you stress the animal or you give them circadian misalignment. So what Lorenzo did, he simply took the animals that either have SCN innervation or lack both SCN and PHB innervation, but both animals lack PHB innervation. And it was quite remarkable. What you could see here is the complexity of these neurons completely is reduced in animals that lack IPRGC input to the PHB. Even the animals that have direct SCN innervation lack the complexity of the VMPFC. And then we looked at the ADT animals that I told you about before, and you could see that this complexity reduction in the apical dendrite of these neurons is dependent on the IPRGC loss. And in fact, if you do a time course analysis, what you notice is that the complexity of these neurons highly is correlated with the loss of IPRGCs in the retina. So this tells us two things. One is that the SCN is not playing a major role in this function. And second, that this effect on the VMPFC can happen both in development and in adult situation. So now that we show this major reduction in this region, we wondered what would happen to functions that are driven by the VMPFC. And the first behavior we did was the free interaction social test, where you put a resident mouse in a cage, and then you give them an intruder mouse. And these two males, if they haven't mated before, they are amicable with each other. So you could see they don't fight like mated mice. They actually try to explore each other's and get close to each other's. However, animals that lack IPRGCs, the ADT animals, what you notice is that they don't like the intruder mouse. And here's the quantification of the data, but really this is best seen as a video. So here's now, I'm gonna show you an ADTA mouse that is the resident mouse of this cage, and Lorenzo's gonna put a wild type animal and see what happens. So this male is sitting in the cage, and now we put another unmated male, but wild type, and you could see that the ADT animals try to do everything to avoid interacting with the intruder mouse. At the beginning, we thought that this is like fear or anxiety. However, when we did many fear and anxiety behavioral on these animals, we didn't see any increase in these behaviors. So we think this is a true social behavior that is inflicting its effect on the ADTA animals. So then we use another test to try to specifically test the ventral medial prefrontal cortex area, because this is, although a visual test, it has been shown by Andy Huberman that there's another component that requires the VMPFC. So let me first tell you about the test. If you give mice an expanding disc and you give them a chance to either escape or shelter in place, what happens when you expand this disc, mice either freeze or shelter. I don't see it very well. Sorry, shelter or freeze. And this is vision dependent and it's not dependent on the MPFC. So as you could see, both wild type and ADT animals do this quite well because the ADTA mice can see. However, there's another behavior that happens in this test, which is called the tail rattling. And people think this tail rattling, which you could see here, is a way for mice to tell other mice that there is a danger in the environment. And as I told you, Andy Huberman's lab showed that this is dependent on the medial prefrontal cortex. And now if you look at the wild type versus the ADT animals, you see a vast difference in the two. Again, indicative that the lack of IPRGCs also affect the function of the medial prefrontal cortex. So finally, I think I have shown you convincingly, hopefully, that this direct pathway is just as important as circadian photoentrainment. And being part of the National Institute of Mental Health, we are very excited now at extending whether this direct pathway is also found in humans. And so we're measuring stuff in the real environment where the subjects are living. And now we have a light sensor to measure the light input to these subjects. But we're also interested in laboratory subject experiments where we're going to measure the pupil constriction to find the threshold for melanopsin responses in a human, as well as fMRI to see if we could find this perihabenular nucleus in the dorsal thalamus of the humans. And because of these studies, we now have a very nice new center in the National Institute of Mental Health in association with other institutes that we call the Rhythms and Blues. And this is really being led by Kathleen Merkangas, and I should add my photo to this group, which I have. And with that, I would like to just acknowledge the people who help us do the work. I failed to mention that the Brain3B experiment was started with Tudor Badia and Jeremy Nathans. Tudor has his own lab now at Romania and the University of Brussels. And a big thank for David Burson and Michelle Ferguson, who really told us to look in the cortex because they had indication that this region may be projecting to the medial prefrontal cortex. And although I didn't show the data, Alfredo Kirkwood for all the LTP, long-term potentiation recording from the slices of the hippocampus. When I was on the outside, the funding agencies, and specifically the Packard Foundation, which allowed me to venture into mood and learning and memory areas. And now I'm very generous to the, I'm very excited or happy with the generous funding of the intramural program here at the National Institute of Health. And I'll be happy to answer any question if anybody has it.